Hello, I'm Stephen Rakusik with SDPB.
And I'm Blaise Rakusik, currently a medical student at the University of Nebraska Medical Center.
If you're wondering, yes, this is my son. Today we're going to dissect the sheep brain. A couple different reasons why we decided to go with the sheep brain. One is it's relatively inexpensive, easy to get a hold of, and the parts are pretty similar to what we find in the human. Some are a little bit different. For the most part, structures are about the same.
Yeah. Yeah. So then a couple things we wanted to mention. When you order these, they may have some kind of ancillary structures that are attached that aren't really part of the brain themselves. So this is a good example here. We have a portion of the nose, the back of some nose structures here that you may see. So if you see what's going on there. And then here we also have some fat and some other tissue and some muscle associated with the back of the eyeball. So that some of these have them, some of them don't. So if you see those, that's what those extra things are on there. Okay, so some of the first things you'll see with the brain here, we have the half brain. It's a little easier to see the layers. So we have the meninges. So you may have heard of like meningitis. So that's an infection or inflammation of the meninges. These are essentially just protective coverings that cover the brain tissue itself. So the most external one, right on the inside of the skull, is going to be the dura mater. So dura for like durable, and so this is like this real thick white tissue that you'll see on the most external portion of the brain. And then below that, we have the arachnoid mater, which is a little harder to see. And actually, it's named so like arachnophobia, like a fear of spiders. It is kind of webby looking. So sometimes when you remove the meninges and when we go to actually pull these off, you might kind of see some stringy webby looking stuff. And that's going to be portion of the arachnoid mater. And then right below that, we have what's called the subarachnoid space, the space right below the arachnoid mater, and that's what is going to have CSF, which is cerebral spinal fluid, which we'll talk a little more about when we get to the ventricles.
Yep.
But that's right below the arachnoid mater. And then the last structure, or the last meningeal layer, is the pia mater, and that is this delicate layer of meninge right on top of the brain tissue itself. So it's this kind of shiny. This one shows it really well right here.
You bet.
It's this kind of shiny layer right on top of the brain tissue itself, and it's essentially attached, right on top of the brain tissue. So it's pretty hard to actually pull that off and like kind of see it. But that's that shininess you see on top is the pia mater, the last layer of the meninges.
How about some of the major structures we see just from looking at the outside of the brain?
Yeah, so when you think of a brain, you think of it's very like lobular and like there's all these like hills and valleys, and so that are the gyri and sulci. So that's actually one difference you'll see between human and sheep brain too, is that the gyri are a lot less prominent on the sheep brain than they are with humans. And so the reason for that is that so the gyri are these like little hills that we see, and the sulci are the valleys. So what those are is like a folding of the tissue. So a way you can think about it is like a piece of paper. So if you have a piece of paper and you kind of like
Try here.
yeah, so if you have a piece of paper and you would scrunch it up, this like hill would be the gyrus, and then the gyri, and then the valleys would be the sulci. And then so while we're talking about those terms, you'll see sulci, gyri, sulcus, gyrus. What that is just plural versus singular. So if you have one hill, that's a gyrus, multiple is gyri. One valley, that's a sulcus, multiple, that's a sulci.
Okay.
And then so the reason for those is that it increases surface area. So imagine you have like this big, if you had the piece of paper, it'd be a lot harder to fit that if it wasn't all crunched up and folded up. You can fit in a much smaller space. So it allows us to fit a lot more brain tissue within the skull. And that's one of the reasons we see, we think of humans having more executive function, decision-making than the sheep would, so that there's a lot more brain tissue that's fit into that spot. And so that's why there's these gyri are so much more prominent on the human brain, which is another difference we'll see between the two.
Absolutely. How about some major structure names here?
Yeah, and then
kind of the prominent ones.
Well, and that actually brings up a good point too. So you'll see the word sulcus and fissure. So we talked about the sulcus, which for those little valleys. Fissure is also a term for like a valley like that, and they can be used interchangeably. And sometimes you'll see that references certain structures names, whatever sulcus, whatever fissure.
Yep.
But in general, a fissure is deeper than a sulcus. So one of the more prominent fissures that we see is the longitudinal fissure, which runs down the center of the brain here, and that's going to separate the right and left halves of the brain. And actually, while we're talking about this hemisphere lateralization, that they call it. There is, so the first thing is that you'll see a lot of crossing over with brain information and structures. So in general, the right side of the brain controls left side of the body, and left side of the brain controls the right side of the body. So like, let's say you have a right-sided stroke in the brain here, you'll see left-sided paresis or left-sided paralysis or like weakness in the body because it's crossed over at that point. And then the last thing with that, there is some lateralization, so there's kind of some specialization for the brain. So for most people, the left side of the brain is kind of dominant side for language. With a smaller subset of people, it's the right side, but so there is some lateralization, some specialization with that.
Nice. How about, like, we see some other structures here, like this, what's this main part here and then the bottom?
Yeah, so in general, if we just kind of want to break it down into big regions, the cerebrum is like when you think of the brain, it's the big lobes. So that's the cerebrum. And then the cerebellum is this little portion in the back, which literally means a little brain. So it's this little brain in the back. And then the brainstem is going to be the attachment point. So you have the spinal cord and then attached to the brainstem, which then attaches to the brain proper.
Absolutely.
So that's the three big regions if we're going to break them up like that.
Okay. And how about different divisions within that cerebrum?
So you can break up the cerebrum into kind of functional regions now. So the frontal lobe is this front portion of the cerebrum, and this has a lot to do with executive function. So what that is referring to is like decision making, complex critical thinking skills, like doing math, things like that. Impulse control. So executive function is frontal lobe. And then also the frontal lobe has a lot of motor function or muscle control. So right at the back of the frontal lobe here, we have the primary motor cortex. And so you can usually find that pretty easily. You have this central sulcus, which is the separation between the frontal lobe and the parietal lobe. So right in front of that, sometimes it's called the precentral gyrus or the primary motor cortex. That is part of the frontal lobe, and that is for muscle control. Then you have the central sulcus, and then that separates frontal and parietal lobe. So in the parietal lobe, right in the front portion of the parietal lobe, sometimes called the post-central gyrus, so right behind that central sulcus, is the primary somatosensory cortex. So that's going to be sensory control, it's like sensory information. And any sort of sensation on the skin or anywhere is going to be all processed here. And then so if we move a little ways back, we still have parietal lobe here. There's also some like spatial awareness control in the parietal lobe. And then the division between the parietal lobe and the occipital lobe is not quite as prominent as between the frontal lobe and the parietal lobe, but the occipital lobe way in the back is so this is the most posterior part of the cerebrum. This has visual information processing. So you'll have information coming from the eyes coming to here, and then there'll be some information that feeds from the occipital lobe to the parietal lobe, and then that's how you get like your kind of visual spatial awareness, because we said spatial stuff with parietal lobe, and that is how that works. And then the last lobe, then we want to talk about.
Just a quick question about that.
Yeah.
So say, for example, like you are playing basketball and you hit your head in the back and you see stars. You hit the back of the head. What's happening there?
So that is would be very indicative of concussion. So what happened there was that you had the force of falling. The back of the brain would have hit the back of the skull, and then that would cause parts of the occipital lobe to fire, making you think you saw light when in fact it was just that tissue being hit. So that's very clinically relevant what's going on there.
Okay.
And then, so then the last lobe we're going to talk about is on the side here. So this side portion is called the temporal lobe. It has a lot to do with, or it deals with hearing, so audio information. And then it also has some memory function. There's some memory structures inside there as well.
And I've heard also for years, if you get hit in that area, it's one of the most dangerous spots on the skull.
Yeah, and that actually brings up, and so what's nice when you're trying to learn all this is that the lobes of the brain so the names for lobes of the brain are the same as the bones that go on top of them. So like the bone on top of the frontal lobe is the frontal bone, and the bone on top of the temporal lobe is the temporal bone. So you're talking about like getting hit in the temple. There's a region over here on the bones where there's like 4 different skull bones that come together in one little suture point. So it's kind of a weak point in the skull itself. And there's actually a fairly prominent artery behind there too. So if you get hit on the side there, that's a fairly common spot for somewhat severe skull fractures and brain injury on that side. So that would be what why that's so dangerous.
Okay. How about we shift to the ventral side now?
Yeah. So
Which ones you got the best here?
this one thing you'll see. So the first thing we're going to look for is a couple of the cranial nerves. So there's 12 cranial nerves. We're only going to look at a couple of them because a lot of the so when you're looking at a brain preparation like this, a lot of them are going to be removed as they're pulling the brain out of the skull. That's because you have all these cranial nerves that are like wires coming out of the ventral side, and then they're going into foramina of the skull, little holes in the skull, so they'll kind of get ripped off. But the two that are usually intact for the most part are 1 and 2. So that's the olfactory nerve, cranial nerve 1, and the optic nerve, cranial nerve 2. But we actually were seeing as we were opening these up, not all of them have nice intact olfactory bulbs. Like, so this one has some really,
Like that for you.
yeah, so this one has really nice intact olfactory bulbs. So that's these little kind of bulby things hanging out in front of here. So these are dealing with smell information. So olfaction is the sense of smell. And you can actually see if we pull up one of those other, yeah, that one right there. So we had said before this thing on front was some of the nasal, the back of the nose stuff, that tissue that was still there. And you can see how this olfactory bulb would be right, involved with all of that. So you have, this actually isn't the olfactory nerve, which can be kind of confusing sometimes. The olfactory nerves are connected to this olfactory bulb, and they'll be removed because they're all intertwined with this nasal tissue, the cribriform plate. But so these, the olfactory nerves are going to be coming down. And it's actually interesting. They're the only nerves in the body that are exposed to the environment. So, and the reason for that is that they're picking up smell information.
Sure.
So you have odorants or chemicals that would give us a sense of smell, and then so those are landing on there and binding to receptors and such. So, and that actually brings up another clinically relevant topic. So with some of the earlier strains of COVID-19, we were seeing that patients were losing the sense of smell. And so since coronaviruses are viruses that infect the respiratory system, we were seeing infection of some of the supporting cells in the nasal structures that were then causing damage and losing. So that's why we're seeing that loss of smell early on.
Okay. And then flip this one up here. This one actually showed a little better here.
Yeah, so
Number 2 then.
now for a cranial nerve number 2, and actually, if we can pull that the other one up again too. So before we were saying we had the back of some of the eye stuff here. So this allows us to kind of see. I'm going to pull back right here. You can see.
I can lift it up for you.
Yeah, pull right there. You can see this little stalk that's running right here. So that'd be the optic nerve. So that's connecting to the eyeball itself.
You can see it right here too, like that.
Yes.
There.
Yeah, so right here you can see it really well where it's coming out the front. So that's the optic nerve that would be going to the back of the eye. And then if you pull back here, we can see where it's running here, and then where it crosses is the optic chiasm. So we said information crosses over the brain, left controls right. So the right optic nerve is going to be crossing over to the left and vice versa. You're not going to have, and I guess one thing I'll mention too, so it's called the optic chiasm. Now we can look at this one here. You can see it well with that. So you'll see it a little better and we'll look at this when we move more of the meninges here. But so chiasm is referring to this crossing or X. So one thing we'll mention is that with visual information, some of it crosses over, some of it doesn't. I think there was a little activity that you're going to post.
Yeah, beyond the support material section on the website, there's a kind of an interesting kind of a lab activity in which you can kind of test the corpus callosum and the optic chiasm, how that information is shared. So definitely check that out.
Yeah. So, and then that's clinically relevant too with some of the strokes in this region. You'll see some interesting visual presentations where you lose sight in certain regions, and it has to do with that crossing over stuff. Yeah. And then I think maybe the last couple things that we can see without actually doing much of without cutting into it much is so we can see part of the brainstem here. So the brainstem is the portion that attaches. So we have the spinal cord down here, and then where that attaches to the brainstem attaches spinal cord to the brain itself. So you can see the different regions of that a little better as we cut into it. But that's maybe the last thing we can see here.
Yeah. Well, thank you so much. So the next video will actually cut into the sheep brain and identify those structures and functions too.